Hi, my name is Tom Lau. I am the director of the Physicians UBI Awareness Center. And one of the claims that's been out there is that some of the cuvettes can actually lyse the blood. Now, this is a cuvette in mind. It has a turbulator in the middle that moves the blood to the surface. It'd be a great concern of anyone's if it was causing blood to lyse and have a hemolysis and have blood in the urine. That certainly is a concern. This is an example of that item, which the blood actually goes and is turned so it's thrown to the outside of the tube where it gets more light, and then it has to hop through this little hub area. Actually an accelerator speeds it up, and then it spins the other direction and actually is thrown again to the walls. A tremendous mixing device. It's a helical mixing system that people who have looked at this have determined that it's very effective as far as moving blood. But you want to be concerned if someone's out there saying blood is being broken apart and then sent back into the patient, that's a problem. And we want to take a look at that. In fact, I went over to Jim, who is a live blood analysis expert and has done thousands of these. And we wanted to look to see, is the blood being broken down? What does the light do to the blood? Maybe we can find that out. So let's take a look what Jim has to say.
Hi, I'm Jim. And we're here today doing what we call live cell analysis. This is a different way to look at the blood. The blood is put on a glass slide and then put under the microscope and projected off a video camera, and then we record this on a DVD player here. And it's just a kind of a different way at looking at the blood and getting a different perspective on the different things in the blood and how it looks.
So we took some of my blood and we put it under the microscope and wanted to see what exactly is happening, and then we'll take a look and see what the light does. So this is my blood. There's a lot of rouleau or coining effect of the blood being clumped together there. It doesn't get a lot of oxygen exchange. So here we have a picture of the fin and the hub. Now realize the blood is mixed with saline, 4 parts saline to 1 part blood. So it's got a lot of movement and room that it can go. And even though it's spinning down this tube, you can just see that it's got a lot of flexibility. Blood does flex quite a bit. Healthy blood does, because of the small capillaries that it's got to go through. So Jim and I took some blood, some whole blood, and we put it with just straight water, just to see what would happen. And what we did see, as you can see on the slide, is a little bit of hemolysis where the blood cells are damaged. Some blood cells would burst, and you end up with then that hemoglobin in the blood and be filtered out by the kidney. So we were kind of anxious to see what would happen. We took the blood and dripped it through at the correct speed, had it mixed with saline, and had it go by the light in a fairly fast manner, just in case that speed would actually cause some bursting. So we kind of, with fear and trepidation, put that next slide on, and it was quite exciting what we saw. All the rouleau is gone. These cells are healthy cells. There's no indication of any burst cells. This blood is going to be able to pick up a lot of oxygen. Okay, so now we've got the one where we high sped through the light and through the machine to show if there's any damage to the red blood cells. Now, what are these other cells we see in there?
The only thing we see, we have them tipped on the sides here, and then you get a little radical cell right there, and those are what they call, they're kind of like a little bottle capped. Those are poikilocytes.
Okay.
Right in here, not very many. Just
the red blood cells look good.
Yeah, the red
no lysing.
Generally, yep, exactly.
Yeah. Okay, so this is our high speed. The light was on, but just to show that the turbulator itself is causing no damage to these blood cells, right? So what do we have? We have someone who's out there saying, a competitor actually, that when you put the blood through this defibrillator, it's going to cause lysing. What do we see? Not really. In fact, we can look through the list of things that does cause lysing, and one of the causes is heparin. One of the causes is if they happen to use distilled water or sterile water instead of saline, or a number of other things that can cause that. I can say personally, after seeing that there are 60,000 of these treatments going on, people are not calling in and saying, hey, we're having a problem. They're calling in saying, this works great. I just wanted to clear the air a little bit and kind of bring to light what's out there and happening in the world of UBI.